So you want to do a science experiment? Well, you've come to the right place. Hello, my name is Marcus King, and today I'll be doing the IJAS how-to video on safety and endorsements. I'll be going over three sections: the introduction, the general safety section, and the biological safety section. The introduction is going to cover the initial concepts of safety in science projects and the endorsement forms. The general safety section is going to cover specialized safety, such as in chemical, radiation, fire, drone, and electrical safety. And the biological safety is going to cover things from human subjects to tissue samples, living things. If you've seen our other videos, you've likely seen tutorials on how to set up your project or how to carry out your experiment. However, this video is going to be a little bit different. I'm going to go over what to do and what not to do in an experiment to keep yourself safe. Again, safety. Science experiments come in a lot of different forms, so this video will cover numerous cases, and I will likely be skimming over some of the more dense information. However, all of our information can be found at our website, ijas.org, and specifically from the Policy and Procedure Manual. So be sure to check that out if you have any more interest or have any specific resources you need to look for. Feel free to pause in the middle of this video if you need to write down something or keep in mind something for later, as I will be moving relatively quickly. I first want to go over the safety contact flowchart, which is the chain of command in terms of safety and endorsements inquiries. It starts at the student level, goes to the teacher level, the regional chair, state safety chair, and finally the SRC, which is the scientific review committee. Each of these levels indicates that one level has to contact the next in order to make another contact with the next. So as a student, you would have to talk to your teacher or your sponsor, who would then talk to your regional chair and the regional safety chair, who would then talk to your state safety chair, and finally, who would communicate with the SRC. Three things for general safety, which will be applicable for all of the safety procedures I'm going to list ahead. You've got to wear appropriate eye protection and clothing. You need to work under a responsible adult. And IJAS has safety sheets you must fill out about your experiment, which include all references to any hazards and protections from said hazards that you must include to make sure that everything you did was safe. We've also got 4 endorsement forms for biological projects, which help exist to help regulate and provide information on the experiment. These forms range from microorganisms to tissue culture. They can all be found at our website again, ijas.org, and they need to be filled out if the project that you are doing deals with any of these concepts whatsoever. So to start off, chemical safety. This is important if your project requires the use of chemicals at all, especially volatile and harmful ones. And what you want to start by doing is you want to review the recommended safety procedures for a chemical. And then, of course, once you're done, dispose of the chemicals according to state and federal regulations. Any project involving any form of alcohol must be verified by the SRC before the experiment, and no project may use prescription or illegal drugs whatsoever. Next is electrical and mechanical safety. This is applicable to any projects that extensively employ electricity or mechanical devices. We often consider the use of 6 to 12 volt apparatuses safer and easier than higher voltage apparatuses. However, if you have an apparatus with 115 volts or higher, they must be in accordance with the National Electrical Code. If they are not, please go see an electrician, because that is quite unsafe. Next is fire and radiation safety. So students using radiation sources like X-rays, high-intensity radio waves, should be adequately protected from them. And of course, this has to be moderated by a responsible adult. No study with any radioactive material is allowed unless it's in a licensed laboratory under a licensed individual, and any student working with burning materials needs to work under a chemical or a fume hood, and wearing face masks to keep out particulates are encouraged. In terms of firearms and explosive safety, you need to contact the SRC if using firearms in any way. The contact must describe the entire procedure in detail and be submitted in enough time for SRC review and approval. The student may also not possess, utilize, or handle any firearm during the conduct of the experiment. All individuals involved in the experiment must wear proper protection and, in case of incident, be behind a ballistic shield. Any individuals operating firearms must be licensed by the Illinois Department of Financial and Professional Regulation as well. So this is a very dangerous set of circumstances if you are doing a firearms and explosive project. Please see the policy and procedure manual for more detailed instructions on how you should continue. Drone safety. Drones may be used in a science project provided the use complies with all federal, state, and community rules, regulations, and ordinances. And again, you essentially have to keep drones to your own territory and your own airspace because drones may not cross into anyone's privacy or airspace in an experiment. Biological safety. Humans have a lot of needs and requirements because they're treated differently than other animals. So again, humans have to read the policy and procedure manual and fill out the endorsement form for the humans as test subjects category. Humans must not be subjected to any harm, and no primary or secondary bacterial cultures can be obtained directly from human subjects or items used by humans whatsoever. Blood or bodily fluids cannot be drawn or taken from humans at all, and any teeth that are used may be sterilized before experimentation. For more detailed instructions, please see the policy and procedure manual. I cannot stress this enough. This video is a how-to, however, it does not cover all of these specific details. So if you are in need of those, please see the manual. Non-human vertebrates. It's a cautionary one to use these. However, it's recommended that if you do, you use small things like plants, bacteria, fungi, protists, worms, snails, or insects that are easier to take care of and dispose of afterwards. However, if need be, of course, the non-human vertebrates have to be in humane conditions. So again, no primary or secondary cultures are allowed, and no intrusive or pain-producing techniques must be used. Chicken or other bird embryo projects are allowed, but the treatment must be discontinued at least 72 hours before scheduled hatch day. At that time, the egg must be destroyed. Microorganisms. In order to obtain microorganisms, students must get a reliable culture from a biological supplier and may not take cultures from any humans, human items, or non-human vertebrates. Again, all microorganism experiments must be conducted in a laboratory setting as well, such as a professional science laboratory or a science classroom. The only experiment involving microorganisms that can be conducted at home is the one with baker's yeast. Any projects involving the growth of mold, rotting organic material, or biofuel cells that use soil must be done in a conducive environment and may not be done at home. And any projects that involve viruses or insect eggs must comply with Health Institute guidelines, and all cultures must be destroyed by methods such as autoclaving before disposal. Please see the policy and procedure manual if you are using microorganisms in your project and you need to know more safety details. Tissue culture. There are a few levels of biosafety that require specific procedures and precautions. Make sure you adhere to those. And tissue culture cell lines must be acquired from a reputable biological supplier, and all experiments must be conducted in a laboratory such as a science classroom or a research facility. Any project involving tissue culture, again, must be done with the help of a professional and should comply with the standards and principles for biological safety. For any special needs or questions about the endorsements, please see the IJAS policy and procedure manual, and furthermore, follow the contact flowchart I presented earlier. I want to thank you. If you want to contact us, our student board email is the studentboardijas@gmail.com. Thank you.